So hi everyone, thank you so much for joining our workshop today on flow cytometry panel design. I'm really excited to have Omar Zid here with us, who is the co-founder of EasyPanel, and he'll be discussing some of the issues that are surrounding panel design and how their automated tool can take out the complexities that come with designing large flow panels. So the EasyPanel software that Omar will be talking about today is also licensed by Proteintech, and it is available on our website as well. So I've included the QR code if you want to check it out and mess with it while Omar is talking about it during this webinar. So before I hand it off to Omar, I just want to share a few company slides introducing Proteintech. So, Proteintech is a leading provider of life science reagents. The company was founded back in 2001, and at that time, it was primarily a supplier of antibodies and immunoassays. And because we have been in the game for over 2 decades, we have a really expansive portfolio that covers about two-thirds of the human proteome. We also offer immunoassays such as ELISA kits and IHC kits, as well as cell separation kits and antibody labeling kits. The company then later expanded to include more product lines. So, for example, we also have our humankind branded cytokines and growth factors. And all our proteins are going to be expressed in human HEC293 cells so that you can be confident that they have the native folding, post translational modifications, and glycosylation that you would expect to see in human proteins. And that ultimately increases their bioactivity compared to proteins that are, for example, expressed in E. coli systems. We also have our Chromatech branded nanobody based reagents. Nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, which makes them ideal for several applications such as immunoprecipitation, immunofluorescence, especially if you're doing things like super resolution microscopy, as well as protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And Protein Tech Genomics manufactures antibodies that are conjugated to oligonucleotides to allow for protein detection alongside your single cell RNA sequencing. So, we are a company that is driven by scientists, and we are here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. For convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We also know the importance of product validation, and we offer open access validation data for all our customers. And whenever we are testing new products, we do validate using things like relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever it's possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology. For example, we have our proprietary antigen specific B cell cloning and engineering platform, which we use to manufacture our recombinant antibodies. Also, we are a global company, so we do have 7 inventory sites, and we also have manufacturing and research sites on 3 continents. So we are able to offer next day delivery to many areas of the world. So, as a company, we measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left shows that over our company history, our products are featured in more and more publications each year. And as of now, we have over 350,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also really quickly want to highlight that as Protein Tech, we strive to be transparent about our products and make their searchability as user friendly and informative as possible. So, we've recently launched some new updates on our website to help our customers search for the right antibody. This includes our unique 3D epitope viewer, which allows you to interact with epitope structures and explore antibody binding sites so you know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need. For example, we have our immunogen region selector toggle, which allows you to filter for antibodies generated from your region of interest. And now you no longer have to filter through all the variations of an antibody from the search results. You can actually view all formats of an antibody, including things like the conjugations and buffer formulations, all on just one page. And the whole reason that we did this was to help streamline your process for antibody selection. And lastly, I just want to highlight that we have also built off the information from our 3D epitope viewer and database, and we've recently launched an AI-powered search tool on our website called Able. So Able allows you to directly chat and ask for product recommendations, help for experimental design, and access relevant validation data, citations, and resources. So for example, you can ask Able something like, can you suggest flow antibodies to isolate T cells from human PBMCs? And Able will be able to tailor a reagents list for you so you don't have to spend hours searching for the right antibodies. So, this AI powered tool is actually the first of its kind, and it was designed with our customers in mind so that you all can focus on your discoveries and not on reagent selection. So, we're really proud to be the first in the industry to launch this. So, if you do have some time, please do check it out. Again, I've included the QR code right here so you can access the bot by scanning that, or you can simply just go onto our website at ptglab.com, and Able lives right next to the search bar, so you can use it that way as well. So with that, I would like to go ahead and introduce our speaker for today, Omar Zid. So Omar is the co-founder of EasyPanel, an automated flow cytometry panel design tool used by scientists at leading biotech and pharmaceutical companies, as well as top universities. And this software also is now licensed by Proteintech. Omar holds a degree in biosciences from INSA Lyon and an MBA from MIT. With a background in immunology and flow cytometry, he founded EasyPanel in 2020, and the company was acquired in 2024 by Dotmatics, which is the parent company of GraphPad Prism, OMIQ, FCS Express, and several other scientific software platforms. So with that, I'll go ahead and pass it off to you, Omar. Thank you so much for being here.
Thank you very much for the introduction, Afrida. Thank you all for being here. So my name is Omar, as Afrida said. I'm the co-founder of EasyPanel, and I'm very pleased to be sharing this webinar with Proteintech, now making EasyPanel, our automated panel builder, accessible to all of you guys through Proteintech's license and website. I'll be doing a two-part presentation today. One, going through some frequently asked questions about panel design and EasyPanel, and then sharing the screen to go through a demo of the actual tool and designing a panel with you guys. So I'll try to address some commonly asked questions which are presented here, including what are the common challenges and considerations when you design a panel, how EasyPanel addresses these for both conventional and spectral cytometry, but then also how do we quantify the quality of a panel, and finally how EasyPanel integrates with flow cytometry reagents offered by Proteintech. So there are quite a few different considerations when you design a panel, right? I think you generally first start by asking a question related to cells or tissues. So you ask a biological question or you formulate a hypothesis that you try to address using this cell biology research technique called flow cytometry. And for that purpose, you need to design a panel, which is where EasyPanel intervenes. That process is generally a bottleneck. The panel design can be quite difficult, especially for larger panels. And that is becoming more and more relevant now with the advent of spectral flow cytometry, or also called cytometry 2.0, the new generation of flow cytometers, allowing scientists to get a lot more data than before. Now panels reaching up to 40, 50, even 55 colors or fluorochromes. But then again, one of the complexities here is panel design, which is where EasyPanel comes in. There are quite a few considerations once you set a given list of markers that you're interested in phenotyping. Which fluorochromes are you going to choose? You need to choose a combination that minimizes spectral overlap, essentially maximizes resolution, maximizes what you're able to read off of each different marker or antigen that you are interested in biologically. So there are quite a few metrics, and again, I'll go through the EasyPanel quality score. But if you're talking about spectral cytometry, you might want to minimize complexity scores and similarity scores and optimize your hotspot matrix. But yeah, generally this mostly boils down to the spillover, I'm using it here as a general term, how much spillover or spread one fluorochrome is having into another. Again, you want to have resolved fluorochromes, resolved dyes, resolved signals, not interfering with each other, so that you're able to get and measure each marker separately with great precision. How you match the markers with the fluorochromes is important. Typically, low expressed markers, meaning markers that are rare or scarce on your cells, need pretty bright fluorochromes, which I think makes sense, right? Because if you put dim fluorochromes on these low expressed markers, you may not detect them with great precision. And inversely, you also want to put highly expressed markers with dimmer fluorochromes in order to not oversaturate the signal that you're detecting. Clone choice is important, can make or break your panel. Antibody clones need to be compatible with digestion and fixation and whatever protocols you're doing when you do the staining. And again, I'll go through that when I share the screen about the panel design itself through the tool, because EasyPanel accounts for that based on a lot of data shared with us by vendors or key opinion leaders or the literature. You also need to account for which buffers you're going to use. Some experimental tips could be very helpful, and again, EasyPanel ventures into these to make sure that your probability of success for your panel is as high as possible. And again, you'll see more all of that in action when I share the screen. So again, just to list all of these, I would also add, besides the expression level of the markers and how these markers are matched to fluorochromes, in terms of brightness, again, low expressed markers matched with bright fluorochromes and vice versa, it is nice to account for co-expression if you know the biology. Not everyone knows how their markers are co-expressed on the different cell types they have in their tubes or in their samples. But if you do, or if you have a sense, then it's especially important to put these co-expressed markers, markers that are simultaneously expressed on the same cell type, on the same cells, on fluorochromes with particularly low spillover. And again, EasyPanel can account for that, as I'll be showing soon. It's obviously important to account for the available lasers and filters, meaning that you need to understand your machine and its capabilities, what lasers it has and which channels it offers for you to be aware of the fluorochromes that you can use. Some fluorochromes may not be used on some machines just because the machines or the instruments or the flow cytometers are not equipped with the optics needed to detect these fluorochromes, specifically, again, the lasers or the optical channels. And again, EasyPanel obviously account for that. It will not suggest fluorochromes that are not compatible with your cytometer. It understands perfectly well your configuration, and the cytometer you're using will be able to suggest fluorochromes that make sense in that regard. Obviously, EasyPanel's magic is that it will take a few seconds or minutes to design a panel rather than long minutes or hours or sometimes days if you're really talking about a large panel. So it breaks that barrier of requiring a lot of expertise when you design a panel, and it saves a lot of time. And last, EasyPanel, because it integrates with the commercial database of flow cytometry reagents offered by Proteintech, makes sure that the products it suggests are actually available off the shelf at Proteintech or other suppliers. Obviously, when you design a panel, you want to make sure you can purchase these products, because otherwise you'll need to do some custom conjugation or you'll need to go through some other complications, which is not ideal. When we design a panel, we try to remain very quantitative, i.e. we've developed hand in hand with flow core managers and key opinion leaders in the field something called the EasyPanel quality score. Now this, while it remains a theoretical score, it's really, really useful and has been used throughout by a lot of flow cytometers to QC or to judge the potential quality of your panel. It's on a scale from 0 to 10. 10 is best, 0 is worst, and it accounts for all the criteria that I just mentioned. What are the fluorochromes that you have chosen as part of your panel? Do they minimize spillover? or similarity or complexity? How are they matched with your markers? Do you have your low expressed markers matched with bright fluorochromes and vice versa? How about co-expression? Are your co-expressed markers matched with fluorochromes with low similarity or spillover, and so on and so forth? So it accounts for all of that, and it's a pretty transparent scoring system, meaning that it really shows you for each of these criteria how much you have scored versus the best possible score. And again, you'll see all of that in action in a few minutes. And like I mentioned, it is important to suggest products that actually exist. Otherwise, things will become really complicated. And for that purpose, we partnered with Proteintech and actually pretty much all major vendors of flow cytometry reagents to make sure that the products we're suggesting are commercially available. And we not only show you the link to the vendors' websites, but also the literature information about these products, whether they're clones or their associated clones, have been published in any of the OMIPs or any of the flow cytometry publications. And again, you'll see these tables showing you the commercial availability of the commercial availabilities of the different products at Proteintech and other places, as well as obviously their prices, the price per unit, and so on and so forth. So with that, I'll be moving along and going through the actual demo of the tool.
So I think you guys can now all see my screen. So the user interface looks like this. Again, it's pretty simple and straightforward. There are 4 steps to generate the panel. Again, a process that generally used to take quite a few hours or sometimes days is now translated into one that takes seconds. First, you start by selecting a cytometer. We have pretty much all cytometers in the market, conventional and spectral, and we're more than happy to add any if any is missing, or whenever any new configurations come up in the market, specifically the spectral ones, we update that as well. So I'm more than happy to take as an example the Aurora 5 laser, one of the dominant spectral flow cytometers out there. It was one of the first spectral cytometers to come into the market. This is its configuration. It's a pretty standard configuration. It is not changed. It is always the same. This is how it looks like. Moving forward, step 2 is where we enter panel requirements. So I'll go ahead and choose species reactivity. We support all species. Human, mouse are the pretty common ones, but if you work on non-human primates or rhesus macaques or what have you, it will be also available. You can specify whether you're fixing the sample and whether you're digesting it, yes or no. And I mentioned this a couple of minutes ago, saying that the algorithm of EasyPanel incorporates knowledge about which antibody clones are susceptible of working or not working in case you fix your sample and in case you digest it. As a personal anecdote, when I have been a scientist, I've been an immunologist
Hey, Omar, I
at
think
the
you got
Curie Institute
muted.
in the past, which is a cancer tissues. I had to digest them. Oh, sorry. Can you hear me okay?
Yeah, now I can hear
Can you
you.
hear me okay now?
Yes.
Okay, sorry about that. Sorry about that. So I was mentioning the fixation and digestion is important to check because some antibody clones will be preferred or excluded based on your choices here. I was trying to give a personal anecdote back when I was a researcher and immunologist at the Curie Institute, which is a cancer research institute in Paris. I used to work on breast cancer tissues and I had to digest them using collagenase. And one of the epitopes that was targeted by one of my antibodies, it was for an immune checkpoint at the time, was compromised. It was destroyed by the collagenase enzyme. So based on knowledge that has been shared with us through the literature or by key opinion leaders, we exclude the clones that are susceptible of not working in case you digest or in case you fix using any of the fixation methods specified here. So let me say no and no here in this case. You can specify the cell populations. So let's say I am interested in CD4 and CD8 T cells, for example. You can exclude fluorochromes, you can exclude channels. Let me move next. And this is the last page before we get to the results. This is where you add the markers. So say I am interested in CD4, CD8, CD45, CD25. You can add them one by one manually. You can also import them automatically as an Excel file if you don't wish to add them one by one manually. We have a very simple Excel file. The template is here. You can download it anytime to your local computer. And it includes the list of markers and the list of fluorochromes. You may have noticed that the expression level is also added by default for the different markers here. CD4 is generally highly expressed on T cells, as you may know. CD45 is also generally highly expressed. CD25 has generally a low expression level. As I explained, this is important and helpful for panel design because low expressed markers like this one should be matched with bright fluorochromes and vice versa. I want to draw your attention to the fact that EasyPanel is a highly automated tool. A lot of things are there set by default, but please feel free to change these if you think they're not relevant to your biology, because after all, you're the one who best knows the biology of your cells. So if you believe in your case that CD8 has a low expression level, please, please, please go ahead and change that, because again, it will impact your panel design. Another really important key takeaway from my presentation today is that EasyPanel is not only a tool to actually design panels from scratch, but also to QC your existing panels. I'm sure that many of the attendees have existing panels that they work with. And if that's the case, please feel free to put the fluorochromes that you're already working with. So let me actually do it on purpose. Say I'm having CD4 APC, I'm using CD8 PE, I'm using a CD25 per CP. I'm actually screwing up on purpose my panel, just because I want to see how EasyPanel's quality score will react to that, the quantitative quality score that I mentioned a few minutes ago. So I said I screwed up my panel, or I'm screwing it up because I'm putting fluorochromes that are not compatible together. For example, PE is a pretty bright fluorochrome, and matched with a highly expressed antigen or marker like CD8 is not ideal. Also, inversely, CD25, a low expressed marker matched with per-CP, a dim fluorochrome is not ideal because we won't be able to detect CD25 very well on our cytometer if we use that panel, if we use that fluorochrome marker combination. So anyways, this is to say that please feel free to put in your existing panels, and EasyPanel will QC it and will give you its theoretical or potential quality score, and it will also suggest what it thinks to be a better alternative if there is. You can also load kits and predefined markers. So say, for any immune cell population, EasyPanel will also be able to suggest a list of markers for you. So say any human or mouse cell population, say I'm interested in human T helper 1 cells. You'll see the list of cell surface markers all listed here based on the add or not, list of transcription factors. EasyPanel is highly automated that is also flexible to adapt to your needs. You can remove some markers, you can add markers. If you're working on a target that is proprietary or that hasn't been described, you can actually write the name of that marker under the marker or the target drop-down menu. This is a free text field, meaning that you can really freely add whatever, not necessarily from the drop-down menu here. So I added this is say an 18 color panel. I added my markers. A few of them have existing fluorochromes. And the last couple points that I'll share here is if you click Show Advanced Options, you'll see the different clones that are commercially available at Proteintech. And the ones in green are the ones that have been published in OMIPS. OMIPS, for those who don't know, are peer-reviewed flow cytometry papers that are highly trusted within the community. The ones, again, highlighted in green are the ones that have been published in OMIPS, and therefore EasyPanel trusts these and will highly likely suggest one of these. Last thing before I move forward to the last page is the co-expression tab. I mentioned that co-expression is one of the important factors in panel design. If you have the information, this is generally the limitation. People don't know what is co-expressed if they never ran this panel. But if you have that information, please feel free to enter your co-expressed markers on the corresponding cell populations. And how EasyPanel will react to that is by attributing these fluorochromes to attributing these co-expressed markers, to fluorochromes with low similarity scores. And what's more, actually for the very common markers that are known to be co-expressed on something, so say, I'll take the example of, I think I had in my panel CD25. Let me add FOXP3 and CD127. These three markers are very well known to be expressed on Tregs, regulatory T cells. So I just added them, and if you go now to co-expression, you'll see that EasyPanel understood that you have Tregs, and it put the three co-expressed markers automatically for you as co-expressed. So it can, let me remove the one in co-expressed group one here. So it can suggest co-expression profiles for the very well-known and common co-expression profiles. Other than that, you'd have to add it. In a few seconds, you'll get your panel design, again, as opposed to a few hours or sometimes days. This is, I think, a 20 or 21 color panel. Generally, we take a lot of time to design, but this is the most interesting page. So I'm calling on your attention. We'll get our panel quality score and the panel suggestion and the commercially available products all in one page and all in a few seconds. All right, looks great. You'll see the EasyPanel quality score being computed here. It won't take more than 10 seconds. And as I explained, it's a scoring system on a scale from 0 to 10. 10 is best, 0 is the worst. And it's a very easy to understand, and it's also a transparent scoring system. Because if you click details, you'll read all the different criteria by which we and experts in flow cytometry judge a panel quality. And we try to quantify this. We try to have a number rather than saying, oh, this is a good or a bad panel. So one of the criteria is, for example, as I mentioned, do you have any low expressed markers matched with dim fluorochromes? Because that's not ideal. And for each factor, you have your score out of the maximum or the best possible score. The whole thing summing up to 10. Some of the other criteria include the complexity index, for example. And for each of these different criteria, you can read more about it here. So that's very cool. Complexity index, the similarity chromes, whether you've accounted for co-expression or not, they're all listed here. And EasyPanel, again, tries to translate that in a score that is easy to understand, even for the non-experts in flow cytometry. Generally, panels that have a score lower than, say, 9 mean that they have some areas for improvement. And that is the case here. Surprise, surprise. You guys remember that, I put per CP on CD25, which is not ideal. And the tool tries to specify or pinpoint to these issues, saying that CD25 has a low expression level matched with per CP, which is a low brightness score on the specific flow cytometer that we chose. And therefore, this is not great. And so it shows the yellow warning sign here. The cool thing is, if you go down, EasyPanel will suggest what it thinks to be the ideal or the ultimate best panel. And it's what is called the fully computed version of the panel. And if you click details, it will show you different criteria, again, by which the panel is assessed. And then if you click view, you'd be able to see what EasyPanel suggests. And again, this is how you can move between your requested panel and the fully generated version of the panel. And so you can see here and check the different, commercial availabilities. If you click view, you'll see its commercial availability, ideally at Proteintech, the catalog number. the price, the list price in the US, and then the link directly redirecting you guys to Proteintech's website for that particular product to find additional information about it. And so this is how you actually choose products. So hopefully that makes sense. In the bottom here, you'll see the different spectral signatures corresponding to the fluorochromes that you picked or that EasyPanel picks on the specific cytometer. In this case, it was the Aurora 5 laser, as you may remember. Further in the bottom, you'll find some gating strategy suggestions. These are copy-pasted images just from the literature in case the tool notices some similarity between the panel that you tried to design and a panel that has been published, i.e. 9 or more common markers. I did mention buffers. This can also make or break your panel. EasyPanel will try to suggest some buffers that you could use based on the fluorochromes and the panel that has been suggested. Now you can save and share that panel through the export buttons and options in the bottom or at the top. You can export it to Excel to your local computer. You can click share if you want to share this panel with your colleagues or integrate it with your lab notebook. You can click share and EasyPanel will generate a unique public link that you can copy and paste again in your electronic lab notebook, in your Teams message, in your email, in your manuscript, in your PowerPoint slides, and anyone who clicks on that link will be able to access that panel exactly as I see it here, even if they've never heard of EasyPanel before. I want to draw your attention to a couple last things. One is, again, if I click under products, you've seen me doing this. Under products, if you click view, you'll be able to see the commercial availability at Proteintech, which is just right here. And every time we actually show the references for these clones. So for example, here you see the vendor catalog number, the different species against which an antibody clone reacts, the clone here, and whether the clone has been published in OMIPS or other publications or not. And each of these is actually a hyperlink. If you click, it will open a new tab showing you that particular paper. The isotype, the size, the vendor recommended quantity or dilution, the price, and the price per unit size or per test. And every time, again, like I mentioned, you have the link to remove that to the specific product on Proteintech's page, which includes some additional information and so on and so forth. So hopefully that all makes sense. The last thing that I'll mention is the ability for you guys to edit panels manually. I think we've been hearing very, very good feedback about this tool. And I was actually there at CYTO, the Global Cytometry Conference, a couple months ago in Denver, and there were a few posters by independent scientists in pharma and academia who designed panels and who actually ran them, conventional and spectral, using EasyPanel. And they were showing the results. They were pretty nice results. But I also want to take a step back as a scientist and acknowledge the limitations behind this algorithm. For example, we do not account for laser intensities. If you're doing conventional cytometry, we do not account for PMTs. We do not account for titrations. So there are limitations in EasyPanel, as is the case for pretty much any other algorithm. And so this is why we give you guys the possibility to edit panels manually. Your experience is highly appreciated. If you believe anything should be edited or changed, feel free to do that. Again, click Edit Panel manually. A new tab will be opened, this time under the Manual Panel Builder. It will show you all the commercial availabilities at Proteintech, and it will let you swap fluorochromes accordingly. This will take 20-25 seconds to load. And like I said, it will let you swap fluorochromes based on your experience. And then once you move forward after your manual edits, you'll be able to get your new updated quality score. So that will be, this will be where I conclude. More than happy to take any questions. Hopefully that's a tool that will facilitate panel design and break this barrier to all of you guys for conventional and spectral cytometry. I think it's highly complementary of the fantastic commercial catalog that Proteintech has, which is again fully incorporated into this tool so that you can design, find, and purchase directly from Proteintech. Open to any questions, obviously, and again, hope that makes sense.
Great. Thanks so much, Omar. Thank you so much for that training and showing us how to use the EasyPanel tool. Before we begin the Q&A, I do just want to share a few more slides. So I'm going to go ahead and share my screen. All righty. Okay. So before we begin the Q&A, I want to share a few slides on some of Proteintech's offerings for flow cytometry to kind of complement Omar's talk on panel building. So in addition to our polyclonal and monoclonal antibodies, Proteintech also offers a wide selection of recombinant antibodies that are validated in and optimized for flow cytometry. So all our recombinant antibody clones are carefully selected after multiple rounds of validation to identify clones with the best signal intensity and lowest background. So this is going to include antibodies from top-sided clones that are produced with rabbit and mouse host isotypes. So you will get the epitope and binding affinity of gold standard clones, but on that recombinant backbone that fits your panel. Our R&D team has also genetically engineered new recombinant antibody clones that have optimal performance in flow cytometry, as well as antibodies that have a mutated Fc receptor binding domain, which eliminates the need for Fc receptor block. These are known as our Fc0 Rab antibodies, and I'll talk a little bit more about those on the next slide. And then finally, we also have our nanobodies for flow cytometry, and these are going to be specifically for immuno-oncology targets. And since these products are all recombinantly manufactured and they rely on cloning into high-yield expression vectors, you get all the benefits of recombinants, such as consistent performance, high lot-to-lot consistency, and unlimited supply. So as I mentioned, I want to talk a little bit more about a really exciting product that we have, which is our FC0 Rab antibodies. So these antibodies are a newer addition to our flow portfolio, and they were designed in order to eliminate background due to Fc receptor binding. So a lot of immune cells, especially things like monocytes, they express Fc receptors. And unfortunately, the antibodies that you use for your flow experiments have Fc receptor binding sites. Therefore, your antibody might be binding to your protein of interest, or hopefully it is binding to your protein of interest, but it also might be binding to the Fc receptors and causing a false positive signal. So to get around this, we generally use an Fc receptor block to mask these binding sites from your antibodies. But Fc receptor block not only lengthens your protocol, but it also doesn't completely get rid of that false signal. And you can see that really well here in this data where we're looking at CD3 and CD4 expression. And you can see that without Fc receptor block, you see the CD3 and CD4 populations. But slightly below, you also see cells that are a little bit lower in level for CD3 and CD4. And this is going to be that background from the Fc receptor binding. So when you add your block, you are going to get rid of some of that background. However, if you utilize the FC0 Rab antibodies, you can see that that background from the Fc receptor binding is virtually gone. And that's because the Fc receptor binding site on these antibodies have actually been mutated. Also, these antibodies, for these antibodies, we use AccuBright site-specific stoichiometric conjugation. And the reason we do this is so that we can control the degree of labeling, brightness, and we can also eliminate that possibility that the fluorophores might block your antigen binding site. So all our FC0 Rab antibodies are also going to use a rabbit IgG backbone so that only one isotype control is needed for your experiment. So it simplifies your experiment a little bit. And they're also going to be combined with top-sided and reliable clones. So we are doing samples of our FC0 Rabs for this webinar for all our registrants. So stay tuned for the last slide. I'll give a QR code where you can request that free sample. So next, I want to talk a little bit about our dyes. So our antibodies for flow cytometry are conjugated to either conventional dyes or our in-house Coralite Plus dyes. So our Coralite dyes are going to be bright fluorescent dyes, which offer high photostability and minimal fluorescent bleed through, which allows you to use several of these dyes in a single panel. And these dyes are going to be equivalent to what you would see in spectral profile to the Alexa Fluor dyes, and they also undergo very stringent validation to ensure reliable results. And then here is just an overview, a chart of all the dyes that we offer that are compatible with several different lasers. So as I mentioned, we have our in-house correlites as well as antibodies that are conjugated to conventional dyes. So we have things like FITC, PE, APC, and then we also have other dyes such as Atlantic blue, which is going to be spectrally similar to Pacific blue, or cardinal red, which is spectrally similar to Texas red. And then for instances when a pre-conjugated primary antibody is not commercially available to you, or the ones that are just don't fit into your panel, we do offer a solution at Proteintech for that, and that is our flexible antibody labeling kits. So flexible, the way that it works is that it uses a high affinity linker, which allows you to label your own antibodies in just 10 minutes, regardless of the antibody supplier or antibody buffer composition. So the way that it works is that you basically take your primary antibody and then you add in your flex linker, and that flex linker is going to have the fluorophores that are going to be covalently attached to it. And then within minutes, that flex linker is going to bind to the FC portion of your primary antibody. You then add in your flex quencher, and that's going to bind to any of the unbound flex linker, and then that's it. You now have a conjugated antibody that's ready to use. And because it's really simple and fast to use, Flexible does allow you to easily rearrange your panel for your multiplex experiments. And then if you are just starting with your panel design, Proteintech also offers pre-optimized flow cytometry panels. These panels, which are listed over here at the bottom, are curated and optimized by our R&D scientists. And the idea is to detect specific subpopulations of cells. So each panel incorporates top-sided clones, and it's designed to have that PE channel open for you so that you can easily incorporate your target of interest into your panel. And then finally, I just want to mention that Proteintech also offers supporting resources, reagents, and kits for flow cytometry, including things like isotype controls, secondaries, viability dyes, cell health assays, and various different buffers. We also offer a wide variety of blogs covering topics from panel design, gating, and data analysis, as well as protocol guides. And we also have all our past webinars on our website from both internal and external speakers. So as I mentioned, as an attendee of today's webinar, you will be receiving 2 follow-up emails. The first one will have a recording of this webinar as well as a link where you can request a free sample of one of these FC0 Rab antibodies from this list. If you want to request your free sample now, you can also scan that QR code and request it right away. And then you also will receive another email that will have a certificate of attendance. So we do ask that you please allow 1 week for these emails to be sent out. And then with that, I just wanted to thank everyone for joining. We'll start the Q&A session. If you do have any questions for us after the Q&A session is over, you can always email us at events@ptglab.com, or I've also put Omar's email right here. And then again, the QR code to access that EasyPanel tool on our website. So let's go ahead. I saw that there were some questions that were coming in. So the first question says, is there a common forum so that users can share their panels?
Yeah, so I'll take this one. Thank you, Afrida, and thank you for the question. In I think in EasyPanel there is a way to different ways of sharing panels, including, as I showed, Excel exports, and then sharing a link that is accessible to anyone if you share it with anyone, redirecting to the panel. In EasyPanel as a tool, because users generally, log in as part of a pharma company or as a university, there may be things related to this. For the case of Proteintech, I think we need to potentially work with the team on addressing that if there is a need for that within the Proteintech flow cytometry community itself. But like I said, a couple options currently existing are exporting panels as an Excel and sharing the link or generating a public unique link and making it accessible to essentially anyone or in your lab notebook. That makes sense.
Great. Thanks, Omar. There's another question that came in that's asking, with the panel score, is it also based on you being able to run the panel without compensation? For example, if you have a score of 8.3, but you include compensation, does it make it better?
Yeah, so that's a good question. I tried to address it in the chat as well. So the score is essentially pertains to how much potential compensation you would have to do as a scientist. The lower the score is, the worse the panel is, and therefore it would mean that you may have to do a lot of compensation in addition to other potential challenges, such as not even being able to detect your markers, especially the low expressed ones, if they're matched with dim fluorochromes. So I wouldn't, the compensation per se won't affect the quality score. The quality score is indicative theoretically or potentially of the general quality of the panel. And again, the lower it is, the more potential compensation you'd have to do. Like I said, I tried to be very specific here and say this is a theoretical quality score. It is a very useful score. But again, it's theoretical because there are quite a few things that it doesn't account for that are really, really hard to account for, such as titrations and laser intensities and PMTs in the case of conventional cytometry, which also affect compensation. But a rule of thumb is the lower the score is, the worse the panel is, and the more potential compensation you'd have to do. And generally, you want to have a panel with the lowest possible compensation to be done, if that makes sense.
Yeah, great. Thanks, Omar. A question came in saying, can you change the configuration for the instrument on the software?
So in the case of spectral cytometers, the configurations don't change because they're standards, meaning that users or scientists in labs cannot change them. So in EasyPanel, you cannot change them. In the case of conventional ones, yes, this is configurable through the admins of the tool at Proteintech. So feel free in that case to connect with your Proteintech representative or to request support. Either we will be able to do it for you, generally within the same day, or your Proteintech representative will be able to do it for you if we set them up as an admin. They have an admin dashboard, and there's actually a pretty simple way in the backend to configure, to add lasers, change filters, et cetera, et cetera. Having said that, we try to include as many as possible of the conventional standard configurations of conventional cytometers. So hopefully that covers most cases and most needs. If you really have a different cytometer, again, you have these two options.
Awesome. There's another question asking, can you enter multiple cell types at one time for a given panel?
You can. So under cell populations, which was in the second page, you can absolutely enter multiple cell populations. Yes, no problem.
Awesome. Okay, it seems like we have one more question, so we'll go ahead with
I can,
and end with this
yeah.
one. Someone's asking how long they can try EasyPanel for. So it does live on our website. I believe each person has like an unlimited amount of use on it. Is that correct, Omar?
Yes. Anyone can go to the Proteintech website. They can play around with the tool for as long as they need it. So totally unlimited. Change panels, edit panels, update panels. Yes, that is all covered by Proteintech. Yes.
Amazing. Awesome. All right, well, if there's no more questions, I just want to thank you again, Omar, for giving us this training and showing us how this tool works to simplify panel design. And I just want to thank everyone again for joining our webinar. And as always, I hope you guys all have the great rest of your morning, afternoon, or evening, depending on where you're tuning in from the world. So take care, everyone.